Hello everyone, welcome to my tutorial. In this video, I am going to show you how to choose the correct protein PDB ID from the protein data bank and how to download the structure from the protein data bank. So the learning objective of this tutorial are as follows. The first one is what is a protein data bank? Here I am going to introduce you with the protein data bank, and then I will explain how to select appropriate protein structure or PDB ID from the protein data bank using different kinds of criteria. And finally, I will introduce you with the PHAROS database, which contain information about the different types of druggable protein targets of different kind of disease. Okay. So first of all, type in Google protein data bank. Then a page will open where you can see the RCSB PDB homepage. Now you have to click here. Then the home page of protein data bank will open. This is the home page of protein data bank, and here is a search page. Here you can type the name of the protein of your interest. So here I am typing the name of hemoglobin. So hemoglobin is a protein which is involved in oxygen transport. Is present in a red blood cell. Okay, so you can see all types of hemoglobin molecules. The structure of all type of hemoglobin molecules are present here. So there are approximately 3,950 numbers of hemoglobin molecules. So this is the list of hemoglobin molecules, and we can change here the number of protein molecules per page. There are I have selected here 25 protein structure per page. You can select it to 50, and now 50 numbers of hemoglobin molecules will be shown here. Okay, so we can refine the search. So you can see here the structure of these hemoglobin molecules have been determined through experimental method. That means X-ray crystallography or NMR or other methods, but not through AI generated method. So you can see here out of 3,950 structures, 1,114 protein structures are from Homo sapiens, that means from human. Okay, so we can refine the search by selecting Homo sapiens and click here. Now only 1,114 protein structures will be shown in this page. Okay, so all these structures are from human. Now then we can also refine the structure or search by selecting the experimental method. Here you see out of this structure 1019 structures have been determined through X-ray diffraction. So it is better to select the experimental method X-ray diffraction. Okay. If the structures are not available from X-ray diffraction, you can also select from electron microscopy or NMR or other methods. Okay, but it's better to choose the structure from X-ray diffraction. Then the most important thing, the resolution. Okay, resolution of the protein structure. So what is resolution? Resolution refers to a measure of the level of detail or clarity in the protein crystal structure. So it is one of the key parameter used to assess the quality of the three-dimensional atomic model. And this resolution is expressed in terms of angstrom. Okay. and lower the Armstrong value means higher the resolution. So it's better to choose the high resolution structure. So here you can see in form 0.5 to 1.0 Armstrong range, only 5 protein structure are available. So I'm selecting here 0.5 to 1 and 1 to 1.5, 1.5 to 2 and 2 to 2.5. That's a considerable number, and I am refining my search. So now only 832 structures are visible in this page. Okay. So in case of choosing appropriate protein structure, always remember you have to choose the structure with high resolution, because high resolution structures are more clear, and these structures have more clarity and more detail. So this is the first criteria for choosing the protein structure. You see, several other criteria are there, and you can shortlist your protein of interest based on the objective of your study. Okay. So now I am selecting this PDBID 7JYP for further exploration. So I have opened this detail of this PDBID here in this page. And the second most important thing or important criteria for selecting a PDBID is the associated paper. So the authors have determined the crystallographic structure of hemoglobin molecule, and you can see they have uploaded 4 different crystallographic structures. So it's better to know why did they determine 4 crystallographic structures. What was the objective of their study? So the objective of their study is to design modulator of hemoglobin molecule. Okay, and they have designed a synthetic inhibitor which is number 23, and they found that this is a potent modulator of hemoglobin. So you should download that crystallographic structure where this number 23 synthetic inhibitor is co-complex. That means they have determined the crystallographic structure of hemoglobin molecule which is bound to the number 23 modulator, synthetic modulator of the hemoglobin. You should download that structure. Okay, because it will help you to compare your drug molecules, the interaction of your drug molecule with that of the interaction of 23 with the hemoglobin molecule. So this complex will act as control for your study. Okay, and also you can get an idea about the key interacting residues which were involved in the interaction with the number 23 synthetic modulator. So you can see the further detail here about this crystallographic structure, and this crystallographic structure have no mutation. That means the hemoglobin molecule is not mutated. The authors have not induced any mutation. That means it's not a mutational study. It's a drug design study, and in this structure there is no mutation. The structure is wild type. So always select the structure which is wild type for particularly for drug design study, because if there is any mutation in the active site residues, the affinity of the protein molecule will change. Okay. Next, you can see this hemoglobin molecule is associated with the heme group and the synthetic modulator and oxygen and other one synthetic molecule. So you will get all detail about the protein structure here, the resolution and other thing, and sometimes the IC50 value of the bound ligand or bound inhibitor. Okay, so sometimes the crystallographic structure of the only protein molecules are available in the protein database. So if your objective of your study is to drug discovery, then I prefer you should download the structure which is bound with any known inhibitor, known synthetic inhibitor. That means the co-complex inhibitor. This known synthetic inhibitor can act as your control in your study. Okay, because you can redock that known synthetic inhibitor in the active site pocket of that protein molecule, and you will ultimately get a docking score. So now you can compare the docking score of your test molecules with that of docking score of the known inhibitor, and it's always a good option. And another thing is that sometimes the only protein, the active site of the only protein is different from that of co-complex protein, because after binding of the ligand, the volume, shape, etc. Is changed in the protein molecule, because there is a theory of induced field, because when the ligand structure change during the binding with the protein molecule, also the active site, shape of the active site changes. So when an inhibitor bind within the active site pocket of protein molecule, the size and shape of the active site changes, and uh, is better to dock the test molecule in that active site pocket, because after crystallization the shape and volume of the active site remain intact, because the molecules become rigid. So in most of the docking software, the flexibility of protein molecule is not considered, only the flexibility of ligand is considered. The protein is like a stiff and stiff molecule, like a stone. So you are docking your ligand molecule within a cavity of a stone. Okay, that's why it's always better to choose the protein ligand complex instead of only protein. Now, after selecting the protein molecule, we have to download the protein structure in PDB format. So now open this link. I am opening the PDB ID. So it's now open, and there is a option download file, and from here we have to download the PDB format, that is protein data bank format. And if I click here, the PDB structure of the protein molecule will be downloaded. So now it's downloaded 7jy3.pdb, and it is present in my downloaded folder. Now I can open this PDB file in visualization software. And for visualization of the PDB structure, the visualization software is needed. Okay, so several visualization softwares are available, and you can download and install. And in my next tutorial, I will show you about the different kinds of visualization software, how to download and install them. And one of the most popular visualization software is Via Discovery Studio, and this software is a free software. You can easily download and install it in your Windows system. It's also available in Linux, Mac, and other versions. Okay, so this is the Biovia Discovery Studio software, where I have opened this protein molecule. So after downloading the protein molecule, you have to visualize the structure of that protein molecule in visualization software. Also, the visualization software have features through which you can process and edit the structure of protein molecules. Because you can see here, the protein molecules contain not only the amino acid but the water molecules. Red dots you can see, these are the water molecules. Different ions which were required for crystallization process. That means different kinds of impurities, bound ligands, and other heterogeneity. So we can remove this water molecule and different ligand or heterogens which are associated with this crystallographic structure. So through this software we can remove this. So after downloading this software, you have to see whether this structure uh, have any missing residues or not. And what is missing residues? So let me show you. So this is a crystallographic structure. Here I am going to show you about the missing residues. So you know the polypeptide chain consists of amino acids, and in this case of PDBID, the first amino acid is glutamine 8. Okay, so this is the amino terminal end where glutamine 8 is present. Now you may question why glutamine 8? There should be one amino acid with number 1. Number 1 to 7 amino acids are missing, but they should be present in the protein structure. So number 1 to 7 amino acids are absent in this crystallographic structure because during crystallization process, this region is a loop region. Sometimes the author uh, can't determine the structure of this region because these loop regions are very thin and it's very tough to determine the electron density map of this region. Sometimes intermediate loop regions also missing residues are present. But the authors know the sequence of the amino acid, but they can't determine the position or coordinates of these residues. Like you see, this chain should be intact, but you can see here lysine 43. This residue is lysine 43. So after lysine 43, there should be 44, 45, 46. Okay, but here again start from lysine 53. You can see this is lysine 53 and this is 43. That means 10 residues are missing here. Okay, so authors have not determined the coordinates of this residue. So actually this polypeptide chain is fragmented. It's not a intact polypeptide chain. So during molecular docking, it will not be problematic because here the protein molecule is a very stiff structure, a rigid structure. It will not move. It is not flexible. But when you will perform molecular dynamic simulation, the flexibility of the protein molecules will also be considered. So during molecular dynamic simulation, the protein molecules will vibrate or move, and during that process, these two fragments may get separated. Okay, so it will be very problematic during molecular dynamic simulation. So always choose the structure which has no missing residue, or if there is no structure without missing residues, you have to add this residue to different softwares are there like modeler for which is usually used for homology modeling. So through modeler software, you can add these missing residues. You can repair this region. Also, PyMOS, Chimera, these softwares are also there through which you can missing residues. But it's better to avoid this structure with missing residues. So after downloading this structure, you should check properly structure of that protein molecule, whether it is intact or it has any missing residue. So it is one of the important criteria for choosing the structure of protein molecules. Okay, I am finally going to introduce you with PHAROS database. So PHAROS database contain the information about druggable protein target. This database is maintained by National Institute of Health. So type in Google PHAROS database and you can see this is the link for opening PHAROS database. So I have opened this link and an enzyme lactate dehydrogenase A I am searching for. So you can see lactate dehydrogenase A within bracket target, associated disease, associated ligand. Okay, so these informations are available. So I am opening the lactate dehydrogenase target. So you can also search for different kind of disease, and this web server will show you the protein target or the protein which could be targeted for drug design for this particular disease. Okay, so this is the page of lactate dehydrogenase A. So the protein summary, the detail about this protein is available. You can see here detail about this protein, and then IDG development level. IDG development level means drug development level. There are four categories, that means T dark, T bio, T chem, and T clean. T dark means if any information, particular information is not available about the protein, then it comes under T dark. If the information, some sort of informations are available about this protein, that means the disease with which this protein is associated, okay, then they comes under T bio category. You can see these are the criteria based on which these proteins are categorized. Okay, PubChem score, or whether any inhibitors are available for this protein molecule or target molecule. If active ligand or synthetic inhibitors are available for this target protein, then it comes under EChem category. That means inhibitors, non-synthetic inhibitors have been reported for this lactate dehydrogenase. But you see, it's not P clean category because there is no approved drug in the market for treating the disease targeting this lactate dehydrogenase. Okay, so if any approved drug is available in the market for any target protein, then this protein comes under this P-clean category. So you can shortlist your target protein based on the reports available or studies available, or if you want to target any protein for which there is no approved drug, then you can select this P-chem category. And if you want to select a protein target which have already approved drug, then you have to start using the T clean category. Okay, now the protein sequence and structure informations are also available. You can see here the PDB IDs of this particular protein, that means lactate dehydrogenase A, are listed here with the PDB ID and their resolution, and there is a link of protein data bank. From here, you can directly navigate to the protein data bank. Suppose this is the PDB ID and I can open it through clicking in the link. Open in new tab. Now the protein data bank is open, and this is the 1I10 PDB ID. So what is the utility of this database? I find this database very useful because if you want to select your target protein, because the best option is literature review. Because if you want to design a drug for a particular disease, the first step is literature review, through which you can identify the different target protein. But sometimes only through literature review it's not possible to identify all the possible drug targets or target protein. So that's why this PHAROS database is there. You can search in the PHAROS database for the target protein for different kind of disease. And these proteins, target proteins have been categorized in different groups. So you can easily find their category, whether any approved drug is available or not, whether any synthetic inhibitors are available or not, and if available, what is their structure. You can see here the synthetic inhibitors of this lactate dehydrogenase. These are the synthetic inhibitors of lactate dehydrogenase which are available, and you can also get the IC50 values of these inhibitors. So this is a very useful database and you can explore more. Several options are there. I find it very useful That's why I'm sharing it with you. Okay. So that's all for today's tutorial. We have discussed about the protein data bank, which is the largest repository of three-dimensional structure of different biomolecules, particularly protein molecules. Then we search the protein data bank for the 3D protein structure, and we discussed about the different criteria through which we can shortlist the protein molecule, or we can choose the protein molecule for our study. And then we downloaded the structure and visualized in visualization software, and we see whether the structure have any mutation or any missing residues, and based on this criteria we discarded the protein molecule. Okay, and we have to select molecules which are appropriate for our study. And then we discuss about the PHAROS database through which we can explore the different druggable protein targets. That's all for today. In next tutorial, I will discuss about the different kind of drug database from where we can download the structure of small molecule chemical compounds. Okay, so that's all for today. Thank you. Thank you for watching.